So the title of my talk is slightly different from what was in the program. Rather than talking about inflammation in general, I will talk about allergic inflammation and discuss some of the, just figure out how this works. Yes.
Next slide, previous slide, and pointer.
Thank you. I think in modern immunology, although there are many remaining puzzles, the puzzle of allergy and allergic responses and type 2 immunity in general, I think, is one of the greatest in that in many areas we understand in molecular details pathways involved in sensing infections, in activating inflammatory responses and adaptive immune responses. But in the case of type 2 immunity, while we have a great deal of understanding of molecular mechanisms operating at the level of T and B cells, there is very little known about the very proximal events in sensing the stimuli that trigger type 2 immunity and allergic inflammatory responses. And there's very little understanding of the biology of allergy in general. So the issue is that type 2 immunity is a specialized form of defense that is mediated by several specialized types of innate immune cells, like mast cells, eosinophils, basophils, mucosal epithelium, ILC2s, and Th2 and Ig antibodies. And the main defense strategy is based on barrier immunity and on expulsion and repair. And it is thought that type 2 immunity evolved primarily to protect against macro and ectoparasites, such as helminths and ticks. And it's thought that allergy is a result of a mistargeted immune response, which is intended for protection against parasites, and that allergic responses to innocuous, non-infectious agents is purely pathological. And that view, I think, is incorrect, or at least incomplete. There are several reasons to think that. There are several problems with the view that all allergic responses are simply mistargeted responses intended to protect from parasites. Although many allergens, such as chitin and many protease allergens, appear to function by mimicking the immunogenic activities of macroparasites, most allergens have nothing to do with worms, nothing obvious at least. Secondly, anaphylactic responses can be extremely rapid. They can operate in the matter of seconds and minutes, and this type of time scale of a response presumably did not evolve to deal with slow replicating macroparasites. Finally, allergic sensitivity is largely mediated by IgE-mediated responses, while IgE is largely dispensable for immunity to most macroparasites, at least in mice. And allergy can develop in response to a huge variety of stimuli that have little in common in terms of their structure or origin. And this includes many examples you're familiar with, from metals like nickel to small chemicals like penicillin to peanuts and latex and so forth. So the alternative view has been proposed a while ago, originally almost 50 years ago, by Higginbotham and colleagues, who demonstrated, this is the first evidence we could find in literature, back in 1965, that mast cells and local resistance against envenomation, so a typical allergic response induced by venoms, in this case snake venoms, was actually beneficial and protective. And the idea that allergy may function to protect the host from noxious environmental substances was first clearly enunciated by Margie Profit in 1991 in this article shown here, which was published in Quarterly Review of Biology, where she clearly enunciated the concept that allergy may have a very beneficial protective role against a variety of toxins and noxious substances that are found in the environment. And that view has been picked up primarily by people who work on evolution of host defenses, but mostly ignored by immunologists and allergists. So allergic host defenses comprise a number of components that all aim to protect the host from something we don't want in our organisms. These include barrier enhancements. Very importantly, it involves removal and expulsion of unwanted substances and macroparasites and ectoparasites. It involves restriction of their spread in the body, involves tissue protection and repair, and importantly, but largely understudied component of these defenses is behavioral avoidance reactions. So all these responses aim to protect us from substances that are noxious in one way or another, or substances that may mimic something that's noxious. And if you look at the list of the mechanisms involved in these defenses, these all entirely correspond with what's known as allergic pathologies. There are two main modules that are involved in allergic immunity. One is mediated by Th2 and IL-C cells, and these orchestrate defenses by eosinophils and by mucosal and skin epithelium. And the second module centers on IgE and mast cells. And these defenses control, through the effect largely of histamine, the effects on vasculature and smooth muscles in airways and in the intestine to mediate a variety of expulsion reactions. This module also involved through histamine-mediated pathway in activating C fibers that are specialized on itch sensing. And in addition, mast cells produce a variety of enzymes that help to detoxify various noxious enzymes found in venoms, for example. And they're involved in expulsion of ectoparasites by unknown mechanisms. In addition, there is a very interesting connection between allergic sensing and neuronal sensing. The mechanisms illustrated here show how detection of noxious substances, for example, in the intestine through enterochromaffin cells, leads to production of serotonin that activates sensory fibers of the vagus nerve, and these are responsible for the reactions, neurogenic reactions that we have to noxious ingested substances, and that includes vomiting and sneezing, or to inhaled noxious substances, and that includes sneezing and diarrhea. I'm sorry, sneezing and coughing and vomiting, diarrhea, respectively. And the mast cell-mediated recognition of antigens leads to induction of serotonin through histamine, and serotonin in rodents leads also to activation of C fibers, including the one that's involved in itching. And these types of pathways play an important role in both expulsion and avoidance of noxious substances. And they're presumably also responsible for the phenomenon of conditional allergy, when a neutral stimulus, when coupled with allergenic stimulus, can elicit an allergic reaction. It's a phenomenon that's been described about 100 years ago. So immune responses to microbial, non-microbial antigens is what is currently very poorly understood. While we know a lot about pattern recognition involved in detecting various classes of microparasites, bacteria, fungi, and viruses, there is very little known about immunity to non-microbial antigens, and it's thought that these responses are generally non-physiological, and they are the ones that are responsible for things like autoimmunity and allergy. However, some non-microbial, non-self antigens can predictably and reliably trigger immune responses, and so these responses are not idiosyncratic. They're not just function of some specific genetic variations. And the innate immune system perceives these substances as appropriate targets for the immune response. So therefore, we think that the immune response to some non-infectious agents may be intentional, and these are the agents that are either have a noxious activity or they mimic substances with noxious activity. So the idea is that the immune system has evolved mechanisms to sense noxious xenobiotics or noxious substances in general, and to induce immune responses that enhance resistance to the toxic effects of noxious substances through their detoxification, clearance, expulsion, and conditional avoidance. And this type of pathway, we hypothesize exists that is largely uncharacterized. What sort of sensors are involved in detecting noxious substances and how they are coupled with the induction of the immune response. And that's an area of our intense interest currently. So as an example of a noxious substance that is known to trigger a very potent allergic immune response, we started studying animal venoms as a physiologically and evolutionarily very relevant form of environmental challenge. Venoms are a complex mixture of toxins and various enzymes that are introduced into their victims via bites and stings and used by various animals, including reptiles, insects, fish, and mammals, and used by predators to immobilize the prey and also used to protect from predation for defense of territory and defense of food stores. So it's a very important evolutionary strategy. An immune response to venoms is known to include Th2 and IgE, and these are among the first identified classes of allergens, venoms. Portuguese man o' war is one of the first source of anaphylactic reactions. So the question is, what are the mechanisms by which the innate immune system senses venoms and induces Th2 and IgE responses, and what is the purpose of the immune response to venoms? So that's what we started studying. And venoms are complex mixtures, as I mentioned, and one component of venom, called melittin, is a small peptide that we previously showed activates inflammasomes but doesn't play any role in induction of Th2 response. Another component of venom, phospholipase A2, was previously shown to be capable of inducing Th2 response. And importantly, phospholipase A2 is the only known universal component of all venoms from different sources. So if there is a mechanism to detect venoms, and if that mechanism would be targeting an invariant component of venoms, then PLA2 would be a good target from that perspective. So PLA2, of course, cleaves phospholipids, such as phosphatidylcholine, into arachidonic acid and lysophospholipids, like lysophosphatidylcholine. It has a variety of toxic effects on different cell types, and it's a major allergen in bee venom, and it's a major target of the IgE response to bee venom. So bee venom anaphylaxis is presumably mediated by PLA2. In addition, hematophagic fluids, such as one used by ticks, for example, also contain PLA2, and we think it's also responsible for immunogenic activities of hematophagic fluids, which are related evolutionarily to venoms. So bee venom and bee venom phospholipase A2 both can induce Th2 responses, as shown here. This is immunization of mice with bee venom or PLA2 derived from bee venom. Both are potent inducers of Th2 response. In this case, we're using Rich-Loxley's forget mice to measure response directly in situ. And here is a standard re-stimulation following immunization, showing that all the classical type 2 cytokines are produced by T cells following immunization. And immunization with both bee venom and PLA2 from bee venom leads to induction of both IgE and IgG1 antibody responses. In addition, venoms from various snakes, including western diamondback and northern copperhead, also can induce immune responses, as you can see here, and looking at using forget mice as a reporter. And PLA2 from one of the snakes that we could obtain PLA2 from eastern diamondback, can also induce Th2 response. So suggesting that various venoms are immunogenic, induce Th2 response specifically, not the other types of responses, and PLA2 appears to be the common component. So PLA2, of course, has an activity of cleaving of arachidonic acid in the second position of phospholipids and releases lysophosphatidylcholine and arachidonic acid. And the enzymatic activity of PLA2 is necessary for induction of Th2 response, as expected and previously shown. And enzymatic activity would generate two products, as I mentioned, arachidonic acid and lysophosphatidylcholine, or LPC. So we've tested which of them is responsible for Th2-inducing activity, and it turned out to be LPC, as shown here. So LPC could be used together with ovalbumin as an adjuvant, and it functions as type 2 adjuvant to induce Th2 response, and arachidonic acid by itself doesn't. In addition, both insect and snake venoms, in addition to inducing Th2 response in vivo, can activate basophil IL-4 secretion in vitro. And basophils and mast cells are the only two cell types that we could find that would have a direct activity in response to venoms. So this shows that they both can induce this activity, and that LPC specifically is sufficient to trigger IL-4 production from basophils. The response, Th2 response to B venom PLA2 is MyD88 dependent, as shown here, but it's not dependent on IL-1 receptor, on IL-18 receptor, or on various TLRs. Instead, it's dependent on another MyD88 coupled receptor called ST2, which is a receptor for IL-33. And IL-33, as you know, has been previously implicated in type 2 inflammatory response. So what's shown here is that comparison of wild-type and ST2 knockouts and immunization with PLA2, and the response is largely mediated by ST2. The smaller component that is not ST2 dependent, we think, is mediated by, probably by IL-1 alpha, but we're still testing that. Accordingly, response induced by LPC plus ovalbumin is also ST2 dependent. And so this ST2 deficient mice provided by Andrew Mackenzie demonstrated that the part of the response, part of the sensing of the enzymatic activity has to do with LPC, with release of IL-33. And the requirement for MyD88 dependent signaling by ST2 resides in T cells and not in dendritic cells, surprisingly. By looking at conditional MyD88 knockout in T cells versus DCs, we find that defect only is found in T cell specific MyD88 knockout, not in dendritic cell specific MyD88 knockout. And sorry for this, this is a couple of slides that have this problem with conversion, but what this shows is that PLA2, both in vivo and in vitro, can induce IL-33 production. This is IL-33. This is in peritoneum, and this is in vitro. And LPC can do the same. So these slides just show that these substances can induce production of IL-33. Because IL-33 is involved in activation of ILCs, this result demonstrates that PLA2 induced ILC2 activation and activation and IL-5 production is dependent on ST2. And accordingly, eosinophilia also is largely dependent on ST2. So the question then is Th2 IgE response to venoms purely pathologic, or does it have a protective beneficial role? And that's the question that we are very much interested in, and it's actually a very difficult question to address. So what I'll show you is some evidence suggesting that allergic response to venoms can be protective. So what's shown here is that the result of immunization of mice with PLA2, resulting in establishment of anti-PLA2 immunity and subsequent sublethal challenge with PLA2. And the readout that we use here is hypothermia, which is a standard, which is a well-characterized readout to envenomation in rodents. And this is what happens in unimmunized mice. They become severely hypothermic, and mice that have been previously immunized are less hypothermic. And this protective response is mediated by antibodies, because in MUMT mice that lack antibodies, like B cells and antibodies, this protection is eliminated, suggesting it's antibody-mediated response. But when we took serum from immunized mice and transferred it into non-immunized mice, to our surprise, we didn't see transfer of protection, suggesting that it's not a soluble antibody. We thought that it would be IgG1. So that suggested that it's a non-soluble component, which it would be IgE. And we so far tested the role of IgE using Fc epsilon receptor knockout mice. And it illustrates that at least to a degree, this protection was dependent on Fc epsilon receptor and therefore on IgE. So that result demonstrates that IgE-mediated response was protective to some extent in this context. And that is, of course, a very preliminary, but it's sort of a proof of principle evidence that IgE-mediated mast cell activation can be protective in this context. And that, of course, is very much consistent with this previous work from 60s on the protective role of mast cells and more recent work from Steve Galley demonstrating that mast cells play protective role against snake venoms. So conclusions of this part is that PLA2 is a universal component of venoms of different origins. It's a major inducer of Th2 response and a major allergen in various venoms. Its enzymatic activity is required for the induction of Th2 response, and the product of enzymatic activity, lysophosphatidylcholine, is sufficient to induce the same response. And the primary response to PLA2 is largely mediated by IL-33. As I mentioned, there is also possible contribution of other IL-1 family members, presumably mostly IL-1 alpha. And interestingly, it's only primary response that was dependent on IL-33 ST2 pathway. Multiple immunizations, generation of effector memory cells, those responses now become they're ST2 independent. And so there is some interesting uncoupling between primary and secondary response that we currently completely don't understand what the basis for it is, but we're investigating it. And so I showed you some evidence that type 2 response to venoms may promote protection from future envenomation. And it is mediated, at least in part, by Fc epsilon receptor. So we're interpreting this data to suggest that the allergic response to venoms have evolved to provide a protective function, and it's only when this response becomes hyperactive it can lead to allergic pathology such as anaphylactic reaction. So now I'll switch to the second part of my talk, which has to do with analysis of some early proximal events in initiation of Th2 response. And as you know, this issue has been extremely complicated, and it's probably one of the areas where in modern immunology where there are probably more models than in the rest of the immunology combined. Most of these models are not really compatible with each other. And I think that what that indicates is that there are multiple pathways for Th2 response. It's not a single pathway like in the case of Th1 and 17, but there are several different pathways that are completely unrelated to each other, at least at the proximal stages. And while we know a lot of details of what happens right after this sensing occurs, there is still very little understanding about the initial sensing events, and that's what we are interested in. And the story I'll tell you about has to do with the involvement of a particular pathway mediated by a dendritic cell subset for induction of Th2 response. So this is just to point out that we know a lot of details about pathways involved in initiation of type 1 immunity, and very little about early proximal steps that happen here for the induction of Th2 response from naive T cells. And the main difference that we know of currently is that in all other cases, in case of type 1 immunity, dendritic cells are the source of the instructive cytokines that induce T cell differentiation. In the case of Th2 response, we don't know what the source of the cytokines is. And various cell types have been proposed. We've proposed that basophils are an important source of cytokines, others of the signals, but what these signals are is still largely unknown. And again, as I mentioned, this is a very controversial field, and some of the things that I'm saying would be only reflecting my opinion. So we were interested in actually in a very different question initially. We were trying to find a molecular mechanism involved in dendritic cell-mediated tolerization. This was the project that was started by a student about 7 years ago, and we were analyzing what's known as spontaneous maturation of bone marrow-derived dendritic cells. It's a phenomenon that's familiar to everybody who have used these cultures. When you make GM-CSF cultures of dendritic cells, you get the canonical immature dendritic cells, and you always have a fraction of spontaneously mature dendritic cells that express MHC class II and CD80, CD86. And these dendritic cells have been previously looked at primarily by Ira Mellman, who found them to have a tolerogenic role. And Ira's group characterized some of the mechanisms involved in the functioning of these cells. So we were interested in signals that this kind of DCs may produce to induce tolerance. That was our initial interest. And that led us to the story that I'm going to tell you. So we analyzed these dendritic cells from bone marrow cultures and found that, as expected, they express all kinds of markers of mature dendritic cells, including positive and negative co-stimulatory molecules. And when we sorted these dendritic cells and compared them to each other, what we found is that PDL2 molecule was specifically associated with this dendritic cell subset. So we used PDL2 as a marker initially for these cells. And if we sorted dendritic cells based on PDL2 expression, PDL2 negative dendritic cells, which are immature standard dendritic cells, they all respond as expected to TLR ligands, and they make various cytokines. And PDL2 positive dendritic cells, which are these cells, they do not respond to TLR ligands, as you can see here, or they're hyper-responsive. And they don't produce secreted cytokines. And they actually express very low levels of a variety of TLRs with the exception of TLR9, but as I showed you, they don't respond to TLR9 ligands either, at least not by producing inflammatory cytokines. And when we sorted these dendritic cells from bone marrow cultures and injected them into mice, we saw that they specifically induced Th2 response, but not Th1 response. And this is PDL2 negative cells activated by CpG, and as expected, they would induce Th1 and Th17 response, but not Th2. So this was the first indication that these dendritic cells may have something to do with type 2 immunity, which was found, basically wasn't what we were looking for, but this was the finding. And transfer of these dendritic cells induces this Th2 response. This is just showing it in a different way, but not Th17 response, importantly. And when we sorted dendritic cells for PDL2 positive and negative subsets, and when we sorted T cells for naive versus effector memory, and looked at whether they're capable of activating Th2 response in naive versus effector memory, what we found was that in the case of naive T cells, PDL2 positive dendritic cells could induce very strong proliferation, but they did not induce any of the known effector cytokines, either Th1, 17, or Th2. When we used effector memory cells, on the other hand, then these same dendritic cells induce very strong production of type 2 cytokines, but not Th1, Th17 cytokines. So they, in all cases, they're very strong inducers of proliferation, and they're not sufficient to induce naive T cells to differentiate into Th2, indicating that there is an additional signal and additional cell type is necessary to induce that differentiation of naive cells in vitro. And that's a situation that's very different for Th1 and Th17, when you can very easily induce response with appropriately activated dendritic cells. And this also indicated that naive effector memory cells somehow specifically can be activated by these subsets of dendritic cell and not by PDL2 negative dendritic cells. So these subsets of dendritic cells these PDL2 positive dendritic cells also exist in vivo, and the counterpart in vivo of these bone marrow derived dendritic cells is defined by a combination of PDL2 and CD301B. And CD301B is a CTAP lectin called MGL2, which was worked on by Akiko Iwasaki in a lab next door, who generated mice that can ablate dendritic cells expressing this gene. And it turned out that they targeted exactly the same population that we were studying looking at bone marrow cultures. And Akiko's lab has a very complementary story, which is with data obtained by a completely different method. So dendritic cells, this PDL2 dendritic cell from skin draining lymph node, have impaired ability to induce Th1 and Th17 responses compared to PDL2 negative dendritic cells, even though they can induce proliferation. But they do induce Th2 response, as measured here by forget mice, using forget mice. And dendritic cells from the skin, so this is now in vivo derived PDL2 dendritic cells, can also induce effector memory Th2 response. So the summary to this part is that PDL2 positive, CD301B positive dendritic cells in the skin draining lymph node represent in vivo counterpart of bone marrow derived PDL2 disease and preferentially induce effector memory Th2 response ex vivo. When we analyzed gene expression between PDL2 positive and negative bone marrow disease, we found that there are many genes that are differentially expressed, and we spent probably 4 years pursuing genes that were most differentially expressed, and those genes, we specifically focus on transcription factors, and the genes that were most differentially expressed were all kinds of exotic transcription factors that we wasted 4 years studying, and they had nothing to do with this phenomenon. And the transcription factor that turned out to be important was somewhere towards the bottom of the list, and this was a transcription factor IRF4. And IRF4 is preferentially, if not specifically, expressed in PDL2 subset. If we sort by PDL2 CD301, it's almost exclusively expressed in that subset. And this is intracellular staining for IRF4 showing its preferential expression in PDL2 disease. And IRF4, as you know, is a very important transcriptional regulator for a variety of aspects of immune response, including class switch in B cells, shown by Klaus-Rajewski, and differentiation of Th2 cells, shown by Lokov and Takmak, control of type 2 immunity in Tregs, as shown by Sasha Rudensky. And IRF4 is also known to play an important role in development of different dendritic cell subsets, as summarized here. And importantly, 2 recent studies demonstrated that IRF4 in dendritic cells promotes cutaneous dendritic cell migration to lymph nodes, and that IRF4 in pulmonary dendritic cells control Th17 differentiation. So we've focused then on our attention on IRF4 and found that it controls the development of this subset of PDL2 positive dendritic cells. This is in vitro cultures using IRF4 deficient mice. This spontaneously matured population expressing PDL2 is almost completely eliminated in the absence of IRF4. And if we put IRF4 back into this knockout mice, it restores expression. It restores PDL2 expression and expression of other markers, as shown here. It overlaps. If we overexpress it in wild-type cells, so we have more IRF4, then almost all dendritic cells become PDL2 positive. So IRF4 is necessary and sufficient for these subsets of dendritic cell development in vitro from bone marrow cultures. We then performed RNA-seq analysis of wild-type IRF4 negative and PDL2 negative versus PDL2 positive disease. And principal component analysis showed that there was a very, very strong overlap between PDL2 negative dendritic cells and IRF4 deficient dendritic cells as compared to PDL2 positive dendritic cells. So that indicated that a very large fraction of specific genes expressed in PDL2 positive DCs was controlled by IRF4. And so in vivo, IRF4 is also expressed in this dendritic cell subset. So next we used IRF4 flox mice crossed to CLNC Cree mice to delete dendritic cells, and we've looked and verified that deletion was largely dendritic cell restricted, and we've done various controls to confirm that other unintended deletions don't play a role. And what we found that when these mice were immunized with ovalbumin and papain was used as an allergen that is inducing Th2 response, what we found was that in the absence of IRF4 expression in dendritic cells, Th2 response to papain immunization with ovalbumin was diminished. And this is another way to look at this response, looking at forget mice or intracellular staining and frequencies of cells. So it was the same result, that IRF4 expression in dendritic cells was necessary to induce Th2 response to papain. Similarly, IgG1 and IgE responses were also diminished in mice that didn't have IRF4 in dendritic cells, indicating that this dendritic cell population, which is dependent on IRF4, is involved in generating Th2 response. At the same time, Th1 response was intact, and I showed data before Th17 response to this to immunizations we used with CpG was also intact. This is not in contrast to previous studies. This is a different system. We used foot pad immunizations here. And so then we infected mice with Nepostrongylus brasiliensis, which is a nematode that's known to induce a very strong Th2 response, and found that Nepostrongylus induced Th2 immune response was largely dependent on this dendritic cell subset expressing IRF4, and the same was true for immunization with PLA2. PLA2 induced Th2 response was also dependent on these dendritic cells, and antibody response induced by Nepostrongylus was also dependent on that. So the summary is that PDL2 positive CD301B positive dendritic cells appear to be specialized on Th2 responses. They are necessary to induce Th2 and IgG-IgE responses to papain, PLA2, and Nepostrongylus brasiliensis infection, and in Akiko's studies they also found this to be true for alum immunization. They elicit a strong Th2 response in effector memory T cells, suggesting that this subset has some signals that effector memory Th2 cells can respond to. However, these dendritic cells are insufficient to induce Th2 differentiation of naive CD4 T cells in vitro, suggesting that additional signals and their cellular sources are required and absent in vitro cultures where DCs and T cells are used. And this is in contrast to Th1 and Th17 responses, which can be induced by properly activated DCs. So although this study identifies this dendritic cell population as important in inducing Th2 response to a variety of stimuli, it also raises the old question of what is this additional signal that is necessary for naive T cell differentiation into Th2. And I think this highlights that now that we know which dendritic cells are involved, that even these dendritic cells are insufficient to induce that response, and suggests that additional cell type, additional source of signal is necessary to induce the response, or these dendritic cells are not appropriately activated in vitro and something is missing that is normally present in vivo. And we are currently trying to identify that. So finally, I want to acknowledge the students who did all this work. Noah, Pam, and Rachel Rosenstein worked on the allergic responses to venoms. Simon Nish and Yang Gao worked on the PDL2 IR4 story. Xuan Yu is a technician who helped them, and Esther Forshan helped Rachel and Noah to finish their story. As I mentioned, Akiko Iwasaki has a very similar story by using very different experimental system, and her postdoc, Yosuke Kumamoto, they both acknowledged for discussions and sharing the unpublished data. We got RFO knockout mice from Klein Raevsky, ST2 knockouts from Andrew McKenzie, and CD11 secret from Boris Rezes. Thank you very much.